All right, so today we're going to be talking about DNA. DNA, and with this, our objectives is first what we're going to start with is our what for today is that you'll be taking notes on what DNA is and what it looks like. Your how for today is when you are able to describe the nitrogen base pair components of DNA. And so before we get started, what I really want to take a look at first is how DNA got to be discovered. And what we would first start off with is the story of this lady named Rosalind Franklin. Rosalind Franklin was a graduate student who was looking for a job, got a job at a college, and ended up looking into X ray crystallography of different DNA. And so, as she was looking into these X rays, what she was trying to do was capture images of X ray DNA molecules so that she could find DNA's structure. And so when she finally was able to do that, this picture over here on this left corner was the picture that she got. And she began finally saying that she has become the first person to understand what DNA looks like, and that is a helical structure. Helical meaning that it looks like a circle. And whenever she was presenting her data, these two people named Watson and Crick saw her data and recognized that it was not a circle, but it was something called a double helix. When they realized that it was a double helix, they knew right away that they were actually correct. So what they did was they took her idea and her picture and turned it into their own without telling anybody. And in 1953, when they figured all of this out, they began researching a little bit more and finally actually became the Nobel Peace Prize winners of first coming up with DNA and its structure. And so, as we look more into our DNA structure, we obviously know that our monomer for a nucleotide is called, our monomer for a nucleic acid is a nucleotide. And that comes with three different parts. These three parts are a phosphate group, which is right here. This comes with an S group, which is a sugar. And in our case, since we are talking about DNA, we're talking about deoxyribose sugar. And then lastly, a base. So, as we're looking at all of these things, our main what for today is these components and these structures of DNA. So DNA is in the shape of what we now know to be a twisted ladder because of those two people named Watson and Crick. And so, as we look at these DNA molecules, let me write this real quick. Great handwriting. So, as we look at these two molecules, this twisted ladder is over here on this right-hand side, and then as we flatten it out, this ladder looks just like this. And so the sides of this ladder are made up of that deoxyribose sugar and that phosphate group. Here would be a circle up at the top, which was our phosphate group. Just beneath it, still on the side of this ladder, would be a small little molecule of sugar. And then lastly, our base, which is sitting right there. And this is just many, many, many monomers or nucleotides all the way down this ladder. The rungs of this ladder, this middle part right in here, are made up of those DNA bases. And those DNA bases are right here on this left-hand side. We've got adenine, we've got cytosine, guanine, and thymine. Those are our four bases that are all in DNA. So, as we're looking at these bases, there is a rule that we like to follow. And this rule is by the name of this person right here. It is called the Chargaff rule. Chargaff was a man that in 1949, even before we found out that DNA was a helical structure or that little twisted ladder, Chargaff, determined that there were equal parts A and T being the two bases adenine and thymine, and there were equal parts of G and C, or guanine and cytosine. So, if we're looking up here, if we had in one little DNA molecule, if we had 10 adenine, we would know that there were at least 10 thymine. And if there are also equal parts of G and C, we would know that G and C would also have to be equal, but they are not equal to the adenine and thymine. Guanine and cytosine are only equal to each other, so there could be 12 guanine and 12 cytosine. So there are equal parts A and T and equal parts G and C. So continuing to look at what these bases are, we have a quick little mnemonic device to help you guys remember those base pair names. And the way that I like to remember it is the A and the T stand for adenine and guanine, adenine and guanine. And with adenine and guanine, the way that I like to remember it is that apples grow in trees. So apple in the tree, and we've got guanine and cytosine down here, but the way that I like to remember that one is cars go in the garage. Cars in the garage. So quick little mnemonic devices to tell us these base pair names of adenine and thymine, guanine and cytosine. And here are those names again, just in case you need them. But again, adenine and thymine go together. Apple in the tree. Car goes in the garage. So, again, these base pairings are coming together. And if we have our phosphate molecule up here, our sugar molecule right here, and our base pair sitting right here, or one of our bases, we've got the base adenine sitting right here, we know that it has to be paired to something called thymine on the other side. One thing that I want to point out here is that we have to get these two molecules to combine together, to bond together. And those bonds that form in the middle of these two molecules would be something called hydrogen bonds. So these are held together by hydrogen bonds. Again, if we look at this one side over here would just be one nucleotide. And again, they're held together by hydrogen bonds right there in the middle. So, one more thing about DNA is that DNA runs in a direction called anti-parallel. Anti meaning against, parallel obviously referring to that math term of running along the same direction. The reason that we have, or the reason that we call this anti-parallel is because one side is running in a 5 prime, what we call it, to a 3 prime direction, 5 prime to 3 prime, and the opposite side of that twisted ladder is running from a 3 prime up here at the top to a 5 prime down here at the bottom. So running in the exact opposite direction as they're going. And again, we call that anti-parallel. That makes it anti-parallel. So if we follow this one over here, this 5 prime side, as we follow it all the way along, this one reaches all the way back over here to this 3 prime side. And then this 5 prime side here at the bottom runs all the way anti-parallel against the parallel to a 3 prime. And one more time looking at this, again, if we're looking at these separate molecules, this right here that I am circling would be considered one nucleotide. We've got a phosphate, a sugar, and its base. And again, as we're talking about that side of the ladder, this whole phosphate and sugar would be one side of the ladder, with our rings of the ladder being right here in the middle. This one looks upside down because, again, it is upside down. We've got the 5' side on this side running in this direction, and on this other side, the 5' side is running in this upwards direction. So again, these ones would be upside down, and we've got a 5' down to a 3', and a 3' down to a 5'. So all anti-parallel. As far as our notes go today, this is a basics of our DNA and our DNA components. Knowing three main things in this lesson would be that we've got 4 bases, and knowing all 4 of those bases, and then knowing that DNA is anti-parallel, meaning that one side is going to go in the upward direction and the other side is going to go in a downward direction from 5' to 3', and then 3' to 5'.